Hello and welcome to all the delegates participating in this seminar on our product Boroflex, which is meant for relieving chronic joint pains. And today we have with us the emeritus workers, researchers in this team in the form of Dr. John M. Hunter. I would like to introduce to you all on Dr. John Hunter's background. Since 1999, when VDF Futureceuticals was formally incorporated, Mr. John Hunter has held consecutive positions of Vice President, General Manager, Executive Vice President, and currently Executive Vice President Emeritus. Early in his career, he wrote and subsequently expanded Futureceuticals' extensive legal library. Mr. Hunter served as Senior Strategic Hub for Research and Development, Legal Affairs, Sales and Marketing. His area of emphasis included invention, clinical study designs and execution, product and claims development, and the ideation, prosecution, and commercialization of intellectual property. Mr. Hunter initiated the concept of strategic partnership and IP licensing at Futureceuticals. Today, he functions as a senior advisor to Futureceutical management. He continues to develop market disruptive products and to help maintain working relationships with Futureceuticals' largest customers. When asked about Futureceutical, he says, I wake up every morning. What can we create today? That is the question in mind. From the very beginning, leadership, ownership, teamwork, and trust have been ingrained with DNA of Futureceuticals. We have always gone the extra light year to foster opportunity for our customers, partners, and for our employees. We are forward thinking. If it's happening, we try to be there before it begins. We thrive on cooperation. We exist to build great things with and for our customers. I feel personally blessed and honored to have had the opportunity to lead this remarkable, passionate assemblage of talent. Mr. Hunter currently lives on a ranch in the wine country of Temecula, California, with his wife, two grown children, three horses, five guinea pigs, a dog, and a large Dutch rabbit. With this small introduction, I must say that I had the first-hand opportunity to meet Dr. Hunter 3 years back when we had begun with the concept of Boroflex introduction in Indian market, and this was in Pune. And it was really eye-opener that molecule like boron, how could it do so many versatile functions in our body, and it can relieve so many problems of arthritic origin. Then we have in our panel today Dr. Shushrut Babulkar. I would like to introduce Dr. Babulkar. Dr. Babulkar is MS Orthopedics, MCH Orthopedics from Liverpool, UK. He is the founder member of International OTA, President-elect of Trauma Society of India, Vice President NAILS, National Association of Interlocking Surgeons, Education Officer, AO Trauma, India Task Force, Lower Limb AO Regional. His Chief Center for Joint Reconstruction Surgery, Shushrut Institute of Medical Sciences, a very renowned orthopedician of India. So I would request Dr. Shushrut Babulkar to start the proceedings.
Thank you very much, Dr. Diwan, and it's my great pleasure to have Dr. John Hunter with us. Without wasting much time, I would give my mic to Dr. Hunter to elaborate us on boron as a molecule and what are his thoughts on boron. And after that, of course, and we all are blessed today to have with us doctors, panelists from our country, Dr. Vasisht, as well as Dr. Dilip Shah, who is a great friend of mine. Don't go on our age. I'm much younger than him, but he is a very dear senior friend. So we are blessed to have him as well. So, Dr. John Hunter, it's all yours. Please enlighten us on your thoughts, and we will listen to it, followed by questions that I will fire at you, and you need to stand by them with your horse underneath and guinea pig in your hand.
I'm very happy to do that. Thank you so much, Dr. Dewan, Dr. Bob Carr, for your kind introduction and your hospitality. I would like to thank everyone who is attending this presentation for taking the time out of their precious evening to spend a little time with us to talk about this exciting material. And we all know how precious time can be at the end of a long day of work. Sometimes at 8:30 in the evening, we're still working. So I am blessed by the fact that you have attended, and I will do my best to make your investment of this time well worth it. So without further ado, I would like to begin the presentation. I need to reduce the side pictures here so that I can see the entire screen. Here we go. I think I can do it. There we go. All right. So I'm going to be talking about a material called calciumfructoborate. What makes calciumfructoborate a superior opportunity for joint health? It's a good question, and we will pursue that. Just to give you an idea and manage our expectations, today I'm going to take just a very brief amount of time to tell you who Futureceuticals is. Then we will talk about what exactly is FruitXB, which is our brand name for calciumfructoborate. We'll talk about that a bit. And then we'll explain a bit about the regulatory and toxicology status of the material. We'll touch upon a brief outline of the key benefits. I will also touch upon some of the clinical evidence behind FruitXB. We have quite a bit of it. I can tell you in advance that after this presentation, if people are interested in viewing the full dossier, we do have a dossier that's been compiled of all the chemistry, toxicology, clinical science behind FruitXB, but I'm not going to try and cover that in our short time this evening. Also, I'd give you just a brief glimpse into what we consider to be the edge over competitors. There are some worthy competitors out there in the marketplace today. We think we have some advantages. And also, we'll talk about calciumfructoborate's success in the United States by describing some of the products that it's been incorporated into. So without further ado, why don't we go to the next slide? Thank you. Who is Van Drunen Farms and Futureceuticals? We are a food company that started out as a farming operation just before the American Civil War. So that was a few years before I was born. The Van Drunens came to the United States and started out as farmers growing onions, chives, cilantro. They were truck farmers. You could change the slide here. Thank you. Since that time, and many generations, 6 generations down the line, the company has grown into a major manufacturer who supplies all the multinational food companies, whether it would be Unilever, Kraft, General Mills, General Foods. We also supply large chains of food organizations such as Starbucks. We also supply GNC, Vitamin Shop, on the dietary supplement side, and companies such as Pfizer, Glaxo, etc. on the pharma side. So we have over 160 years of farming expertise. We manufacture, we're one of the largest freeze drying operations in the world. And we do custom manufacturing for food and dietary supplement companies. We have our own independent research laboratories in Irvine, California, and we create custom products for many of our customers. We could go on. Thank you. This is just a map of the world showing some of our locations. As you can see, we're primarily in the Western Hemisphere, but we do have a large production facility in Serbia, and we keep a quality team on the ground in Qingdao, China, because we do buy some food materials from China, and we found that it's very important to have a rigorous quality control regarding anything that comes from China and is imported to the United States and thereafter to our customers. We could go ahead. Thank you. Primary capabilities: freeze drying, drum drying, air drying. We have many technologies, including pouching of capsules, tablets, pieces of fruit and vegetables. But a big thrust since 2000 has been research and development. And we have very world-class analytical chemistry labs, quality labs, and we also have human physiology labs. Please turn. So we have everything from, on the left, branded IP, which one product we'll be talking about today, calciumfructoborate, through branded ingredients, plant extracts, fruit and vegetable, whole fruit and vegetable for food companies. And again, let's keep going. So our approach to science, we have a robust team of PhD researchers, chemists. We specialize in the discovery, development, creation of scientifically validated but plant-based solutions that people have not necessarily known about up until now. We follow where nature takes us, and we only look into fruits, vegetables, and grains. We don't do any research on animal products whatsoever. It makes sense because we started out as farmers. Everything we do comes from the land. And then we develop our discoveries by doing analytical chemistry and also exploratory biology. And finally, we do clinical science to prove functionality of the things that we've discovered within fruits, vegetables, and grains. Please go ahead. This is just a quick glance at a few of our branded ingredients. We don't need to dwell on this. Let's go on to the next page. So, finally, what is Fruit XB calcium fructoborate? Fruit XB calcium fructoborate is a patented, non-animal, vegan compliant, and I emphasize that because I believe that's important for many populations in the world today. I know in the United States, vegan is growing by leaps and bounds. People would prefer not to be ingesting animal products, whether it was for health reasons or whether it is for ethical or religious considerations, but vegan is growing. But it's a vegan compliant mineral complex called calcium fructoborate. It's chemically identical to borocarbohydrates that are found naturally in certain fruits, such as apricots, nuts, legumes, and certain vegetables. Our material is considered nature identical, which means it is manufactured or in fact synthesized, not extracted. And I will go into why we don't extract it from the materials I mentioned here: fruits, nuts, legumes, and vegetables. Please turn. Thank you. Some important distinguishing points about calcium fructoborate. Again, it's vegan compliant. There are no animal products, no chicken sternums, no shark cartilage. It's proven safe both by rigorous toxicology testing as well as the test of time. We have provided over 5 billion servings over the last 9 years to human beings without any reported adverse events. It's water soluble, it's bioavailable, it is supported by science. Our marketing team always calls it 2020. They say 20 studies in 20 years. Actually, many of those studies include chemistry studies where we were working on the structure. We looked at then pilot studies, and finally we took the studies into people. But we've done over 20 published studies, including all of those. I'm going to talk about 3 functional human clinical studies today. There are actually about 6, but I'm going to talk about the most important. So over the past 20 years, here's how we arrived at Fruit XB calcium fructoborate. We had a scientist by the name of Dusan Milkovic who worked for us, and he was fascinated with the element boron. And he believed that boron could be put into a very unique molecular delivery system by bonding it with a other mineral and a sugar. And he chose calcium, and he actually synthesized calcium fructoborate. Well, interestingly, we synthesized it and patented the molecule, and about 2 weeks after we filed our patent, California University, UC Davis, came out with a big paper talking about boron transport in plants. And in it, they described these borates that were bonded to carbohydrates, borocarbohydrates. So we published our patent, or we filed our patent just in time. Otherwise, we would have never obtained the patent because the UC Davis work would have been considered prior art. Nonetheless, we went on to publish chemistry and safety work. Then we published clinical studies for joint health. Then we launched the molecule as Fruit XB to the US market. And we supported that market with fast acting as well as long-term joint benefits claims. And we provided over 5 billion servings. So that's a very quick and breathless description of a long journey. You could turn the page there. Thank you. So I'm going to talk about the chemistry of calcium fructoborate. It is a molecule. It's not a blend. It is a molecule held together by chemical bonds. It's approximately by weight 91% of a fructose moiety, approximately 3% boron, and about 6% calcium by weight. It exists in kind of a pH-dependent homeostasis between mono and diesters, meaning sometimes it's in a mono presentation, sometimes it's in a di presentation. When it's carefully produced by our manufacturing process, the diester is the predominant majority molecule, and the diester has actually been the one that has been shown to have the joint health benefits. It's the preferred presentation. The monoester tends to manifest in more basic pHs and a higher pH, but in physiological pHs, the diester is the preferred 1, and it tends to be happier in a lower pH. You can change. Thank you. So we spent years studying this molecule. At the recommended serving size of 216 milligrams per day in a single dose, calcium fructoborate delivers about 197 milligrams of fructose. I want to mention that it's not exactly fructose because it has donated hydrogen for the bond and so on. It's fructose moiety, but I wanted to mention it because some people have raised their hand and said, well, fructose, is that good for, type 1 or type 2 diabetics? And our blood chemistry data have demonstrated from our clinical studies, it has zero impact upon blood sugar levels among everyone, including any diabetics. Go ahead. Also at the serving size of 216 milligrams, calcium fructoborate contains approximately 13 milligrams of calcium. So it's not a calcium product. It's not a significant source of calcium. Go ahead. And at a serving size of 216 milligrams, calcium fructoborate contains about 6 milligrams of boron, again by molecular weight. Thank you. So something that I wanted to bring out, should calcium fructoborate actually be considered as a delivery source of boron simply because it contains a boron atom? And philosophically, I'd like you to entertain the thought, do we typically describe water, H2O, as being a source of hydrogen or a source of oxygen? Usually we just consider it as a unique water molecule. Another example, as you all know, as scientists, widely different organic molecules throughout a wide spectrum of possibilities with very different activities contain nitrogen atoms. But do we generally describe them as being a source of nitrogen or being nitrogen products? Or do we concern ourselves generally that these nitrogen-containing molecules might somehow induce nitrogen toxicity? Although we all know if we sit in a room and breathe pure nitrogen, it would be very toxic. Please turn the page. Thank you. The reason I bring that up is that calcium fructoborate is a unique molecule. Now, to date, boron itself has not been listed as what is considered an essential mineral. It's a trace mineral. And as you all know, in certain territories, daily boron limits have been defined. The World Health Organization estimates that people generally consume maybe around 20 milligrams of boron total in their diets every day. In Japan, boron is considered kind of a dangerous element. It's been used as an insecticide. However, as I was alluding to earlier, not all boron-containing molecules are alike. Unlike, for example, sodium tetraborate, which is basically boric acid, or other inorganic sources of boron, calcium fructoborate does not deliver free boron to our body or free boric acid. That makes calcium fructoborate very unique in this regard. Go ahead. So it goes well beyond being merely a source of boron. Due to the higher association constants that hold the molecule together and other bonding chemistries, calcium fructoborate is not really primarily a source of free boron. It's actually a source of sugar borate anions. When calcium fructoborate hits the acid bath in our stomach, a chemistry occurs wherein sugar borate anions, as they move through the gut, are moved into the bloodstream. And we've actually measured sugar borate anions circulating in the blood rather than any free boric acid. I thought that was an important thing to point out. You can turn the page. Indeed, this boron fructose combination is what contributes to calcium fructoborate's bioactivity. And we believe these anions hold the key to its superior activity as they circulate throughout our bloodstreams. And they've been circulating in our bloodstreams for millennia due to our diets. And I'll explain that. Please turn the page. We have historically ingested borocarbohydrates from vegetable sources. Plants take up boric acid from the soil. Their root systems convert it into sugar borates, and it's passed up through the plant and utilized to maintain cell structure and other processes within the plants. So we have always consumed minor amounts of these borocarbohydrates. None of us go out in the backyard or out in the field and pick up a handful of dirt and eat it. But the plants do. They take boric acid up from the soil and they convert it to the borocarbohydrates. Our scientists were the first to discover and patent these borocarbohydrates. And we hypothesized that borocarbohydrates could actually support a variety of health benefits in mammals, including joint health. Then we set about to prove it. Please turn the page. Thank you. So it's a result of research and development many years. Now, because these borocarbohydrates exist in very low concentrations in apples or apricots or certain nuts, etc., extracting these compounds would have been very impractical from a commercial standpoint. We would have to use tons and tons of the source materials. It would be unethical because it would be wasting food, and it would be a waste of energy as well. So we found in a very simple process, it was much easier to simply emulate these molecules identically by creating a synthetic way to produce them on an industrial scale. So our processes make it possible to generate and consume higher levels of the very same molecule found in nature, and to be able to exploit the clinically demonstrated health benefits of this plant mineral complex. Go ahead. So from a regulatory and tox standpoint, it's considered grass in the United States, generally regarded as safe. It's been approved recently as a novel food in the EU. Does not carry any health claims in the EU, but it's considered safe and is being included now in some early products. It's cleared by FSSAI in India. Actually, calcium fructoborate is interesting because it's a chelate of boron that qualifies it for the FSSAI approval, but it's also a salt of calcium. So it's a combination molecule that contains calcium, boron, and fructose. It's fully supported by published peer-reviewed toxicology studies. We have 90-day studies on rats. We have mutagenicity and mononucleus type studies. They're all published and available for review. It's chemically very well defined and quantifiable, and we have defined consistent ways of measuring it with analytical methods that are ISO validated and certified. Go ahead. During a 90-day toxicology study on rats, and also 2 genotoxicity studies. No observed adverse effect levels, meaning the NOEL, for Fruit XB up to 1,161 and 1,171 in males and females, milligrams per kilograms body weight per day, which for a 60-kilogram person translates about to 66 grams per day. And of course, we're not recommending 66 grams, we're recommending 216 milligrams. Bacterial mutagenicity studies and micronucleus tests demonstrated that no mutagenicity or toxicity was evident. We can turn the page. I've certainly demonstrated that we have a safety profile here. Thank you. So we'll talk about the clinical effects briefly. Please turn. I want to talk just for a moment about Womack and McGill. As professionals in orthopedics, I'm sure you're all aware of these tools, but these are considered to be the gold standard assessment tools for joint mobility and joint comfort. The Womack score, the Western Ontario McMaster University's Arthritis Index, I'm sure you're familiar with that, and also the McGill, which is a McGill Pain Questionnaire, or MPQ. It's composed of 78 words that patients choose from to describe their pain. We like both of these scales because they're well published and well validated, and we have used them extensively in our studies. Not unlike other products that are on the market out there. It's become kind of the de rigueur, shall we say. Can turn the page. So I'm going to talk about 3 studies today. Our first study was sort of a pilot that measured short-term intake of calcium fructoborate that improves Womack and McGill scores. The second was a larger study measuring the same short-term intake on 7 and 14 days of calcium fructoborate on Womack and McGill scores. And the third is a subchronic study that's a larger study, again, on 90 days supplementation on Womack and McGill and other objective parameters. Now, on all these studies, we also did blood chemistries, typical hepatic and renal type markers, cardio markers. We also looked at things like IL-1, IL-6, IL-13, CRP on occasion. And we did notice changes in certain inflammatory markers, and I'll mention those a little bit later as well. So we could turn and start a little more deep discussion here. So study 1 was a pilot. It only had 20 subjects, so that would have been 10 subjects per group, double-blind, placebo-controlled. Subjects were confirmed by CT scan, age range 45 to 65 years. It lasted 14 days, placebo against Fruit XB twice per day. We can change the page here. As you can see, both at 7 and at 14 days, we saw both improved mobility. These were statistically significant, and improved comfort, meaning reductions of pain, both at 7 and 14 days. That was very encouraging. So we went on to study 2. Go ahead. In study 2, we had 60 groups, or 60 subjects, with 30 subjects per group in 2 groups. With an age range of 35 to 65, our average age was 50. We used the McGill pain scores as an intake criteria. People with elevated scores between 35 and 60 were allowed in the study. Double-blind, placebo-controlled over 14 days. Same dosing regimen, twice per day at 108 milligrams. Go ahead, change the page. And again, we saw at 7 and 14 days improved mobility with WOMAC and improved comfort with McGill. Both significant, 21% at McGill at 7, 27% McGill at day 14. Very nice, statistically significant scores. So people were asking us, well, you're checking for twice per day or a BID regimen. Our patients would really like to just take it once a day. And we thought that was a pretty good idea. So let's go to the next study. And here we increased it to 40 subjects per group, but we had 3 groups. We had a greater than 50 McGill score as an inclusion criteria with an age range of 35 to 65 years of age. But this study went for 90 days. And we had a placebo. We had a group where people got calcium fructoborate twice per day at 108 milligrams. We also had a group where they only got it once per day at 216 milligrams, once per day in the morning. And we looked at it across 90 days. And here's what we saw. You can turn the page. We had beautiful results for both of the calcium fructoborate groups. We achieved significance in joint stiffness, meaning the WOMAC scores, at day 14 for both groups compared to placebo. If you notice, both of the calcium fructoborate groups started at a higher baseline, so they had a little catch-up work to do on the WOMAC score there. But on the McGill, we achieved significance in pain reduction at day 7. And what's really exciting, we thought this was just wonderful. We saw the downward slope throughout the 90 days at each testing period, 30, 60, 90. Whereas the placebo group, it looked like there may have been an initial 14-day placebo effect, but thereafter it disappeared or it flatlined. So here we showed that calcium fructoborate continues to work over time throughout 90 days. So you take it, it works quickly, and it continues to work. What we saw was 29% improvement in WOMAC index at just 14 days. I think I've mentioned all of these, so I don't want to dwell on it. What we did see was a 19% increase in blood levels of endogenous vitamin D in 7 days. We also saw reductions, 37% reduction in CRP in 14 days. We saw IL-1 reduced by 29% over 30 days. We noticed MCP-1 reduced at 30 days. We also noticed a slight reduction in sedimentation rate at 14 days. And we noticed some changes in fibrinogen as well at 14 days. We don't really dwell on these because we still consider these to be preliminary observations, but they are giving us indications about the anti-inflammatory effects of our material. And we are vociferously researching that as we speak. You can turn the page again. So how does it stand out in the crowd? I want to move very quickly because I know time is flying here and I don't want to impinge upon your time too much. Please turn the page. We've compared it to collagen, the UC2 product that's out there. We've also compared it to glucosamine and chondroitin. We all know glucosamine and chondroitin has been popular for years. But recent studies have been very mixed about it. Sometimes it seems to work, sometimes it seems not to work. Some people are big fans of glucosamine, chondroitin, others think it's a bunch of hoo-ha. I am neutral about it. UC2 is a very fine product. It's been on the market for a while, but it is an animal product made from chicken sternums. If you compare key results. This is honestly not a totally fair comparison because these materials have not been studied in the same study under the same experimental conditions. So we're only comparing reported study results. But nonetheless, over time, we've been able to show FruitXB seems to have a faster results. We know glucosamine and chondroitin, if they work at all, are more like the 60 and 90 day type results. FruitXB is closer to day 7 and it keeps working. UC2 has reported results at 30, 60, 90, 150 days. FruitXB is less expensive than UC2. And of course, if you compare dosages across the bottom, you can see that there are gram quantities of glucosamine chondroitin compared to the lower quantity with FruitXB or UC2. You can turn the page here. Advantages. We're vegan, non-animal. It's fast acting with 7 days and continues to improve over 90. We've been researching it for 20 years. Small dosage and a lower cost of therapy. Go ahead. Why don't we just change the page here again? We've sold over 1 million kilos of this material in the United States alone. Go ahead and turn. The most famous product that has been included in, it's the hero ingredient in Move Free by Reckitt Benckiser, a multinational company, and we have sold them many, many, many thousands of kilos in their product. It's like the #2 joint health product here in the United States. Go ahead. So I'm not going to repeat all of these. You've seen these. I've said them a couple of times. I'd like to thank you for letting me give you this quick and breathless presentation. I'm certainly happy to try and field any questions in the time that we have. And if you have further questions, the experts at Zuventus certainly can help out. I'm always available as well for calls.
Okay,
Thank
at the
you.
outset, let me quickly ask John, we are running short of time. We have only 10 minutes left, and we want to
Yes.
gather as much information from you as possible. John, quickly asking you, what's the onset of action of calcium fructoborate? You said that it is 7 days. So how did you quantify these 7 or 8 days, if I can ask?
Right. Well, the 7-day mark was one of the litmus tests, one of the benchmarks in our clinical study work. And so using Womack and McGill, which are these self-reported subjective studies that are well published and validated, the study subjects had to take these exams reporting how they felt. And we always measured compared to placebo and compared to baseline. And so at 7 days, we have been able to measure in double-digit percentage improvements in reported joint flexibility and comfort from the actual study participants. Anecdotally, I can tell you that we have people tell us they feel results in as little as 3 days.
Yeah, so their patients coming back to you, suggesting that they are subjectively better, they are feeling better, the pain is less, and they are
Yes.
able to walk better without any wobble or pain, and their distance has increased. Is that also been recorded in the data which was captured, John?
Yes, it is. And in fact, in the subscales of our 90-day study, these Womack and McGill questions involve like, I can get up from a seated position better. I feel better when I'm sleeping in bed. My joints feel better. Many, many criteria of joint flexibility and movement. And so we are in the process of actually publishing another paper that measures the subdomains within these assessment tools.
Okay, so based on whatever data you have captured, John, how long do you think we can continue using them? Because we have seen glucosamine sulfate, and you have pitched it against that, and we have had patients who buy this off the shelf and continue for 1 year or 2 years. As far as your molecule is concerned, how long do you recommend it within safety limits that a patient can continue that on recommendation number one? And if by chance, accidentally, patient continues that on their own, have you seen any side effects of long-term use?
I see at the recommended serving amount of 216 milligrams per day, compared to what we saw in our toxicology studies, I see no problem with someone incorporating this into their lifestyle. I would consider this similar to taking a multivitamin. We take a multivitamin every day for life, basically. And I've been taking calcium fructoborate for the last 17 years on a daily basis. No problems whatsoever. Now, I wouldn't recommend taking, 10 times the recommended dosage, but at the serving amount, we believe that this needs to be a long-term kind of chronic use. It's not a cure, it's a support for it won't remove the condition, it ameliorates or slows the condition. Makes it feel.
Any suggestions for those who are prone to develop kidney stones, renal stones, urinary stones? Have you seen anything in them?
No, and there's no chemical indication. There are no components within it that would cause that. We've seen no drug interactions. The biggest clinical study that we have are the 5 billion servings that have been given to people on the marketplace. And there have been no adverse event reports, and people are taking heart medications, people are taking insulin, they're taking sulfonylureas, taking all sorts of different drugs out there, and some people are taking Prozac. We've seen no drug interactions at all.
Okay, so what's the biochemical toxic level that you would want us to look for, that okay, now this is reached, take no more in a particular patient? Any suggestions on that, John?
Well, it's not a titratable type therapy. Like certain therapies, you would begin at a low dose and titrate in, but this is a very low serving amount anyway. So the way to do it, in my opinion, would be to just simply begin administering it and ask the patient to take it for 90 days and have them come in for an assessment. Ask them how they're feeling. You could do a blood chemistry, look for, liver, kidney enzymes and so on. And I will assure you, you'll see no changes in a deleterious manner to their blood chemistries. They'll be fine.
Oh, that's great. Thank you very much, John. With this note, I think Dr. Dilip Shah has used it in some of his patients. So we have only 5 minutes left. So Dr. Dilip Shah, do you want to suggest if you have had similar results in terms of patients' response?
Definitely. Definitely. Can I share my screen so that I can show you in a short time, which I'm having? Somebody has to stop sharing the screen. Minal, are you sharing the screen?
I think Dr. Dilip Shah, let's talk more than sharing the slides, because we have only 4 minutes left. So just tell us what you feel about the molecule.
Yeah. First of all, thank you very much to be a part of this wonderful meeting. And what I want to say is I've used from the time I met Mr. John Hunter, and I was quite impressed by the good effects, because most of my patients are vegetarian and they had a lot of problems about using chondroitin sulfate and many other products. So most of my patients became very happy. As he said, at the end of 7 days, they were quite happy. And then later on, I asked them to take it for 2 to 3 months, because at that time it was not standardized that how long are we going to do it. So I told them, if you can take it for 2 or 3 months, then it's going to be very nice. So this is how they went on taking 216 milligrams per day. Now, there was little ambiguity because I saw there were some people who insisted taking it on empty stomach early in the morning. And then there were some people who were taking after meals, being a food supplement. So I will just ask a short question to John Hunter. Would you prefer on empty stomach or post meal, so that everybody understands well? And then I'll continue with my approach.
Yes, I would recommend taking it first thing in the morning, a half hour before breakfast. That's what we generally instruct everybody to do. It won't ruin the effect if people take it in the afternoon, but it's best.
Thank you very much, and I'll continue because we have shortage of time. So I have around 100 patients. I have treated from practically 3 years or so, and they have been very happy. Only thing is the cost was the constraint, and most of them were buying from Amazon. And now that we have from Zuventus, it will be available. Yeah. So they will not have much of a difficulty.
Okay.
So
my next question to Dr. Vasisht. Dr. Vasisht, in India, we believe that most of the pain that our Indian patients get is because of osteoporosis and osteomalacia existing together, and these patients developing microfractures, and that's why acute exacerbation of OA. Do you think with this talk of Dr. John and experience of our respected friend, Dr. Dilip Shah, you in North India, where it is predominantly seen, and in Gujarat, part of Gujarat, where we see a lot of genuine varus deformity than the rest of the country, do you think, Dr. Vasisht, your take on this, on use of this molecule? Please unmute yourself. Unmute yourself, Dr. Vasisht.
Okay. Yes, so Shudya, it's very pertinent question you are asking, because most of our patients in orthopedics, they are like osteoporosis, osteomalacia plus osteoarthritis. Most of our problems are, there are very few patients whom it's, end of the day, what we have been discussing. I was also introduced to Mr. Hunter 3 years ago in Delhi. So I was, refreshing that presentation actually. And I also had got few samples for use, and then used it, and then it was not available actually. So it's a micronutrient. Most you can stretch your this thing to say antioxidant. Then till here it's no problem. So if you try and extend it scientifically, then even like Mr. Hunter also pointed out, it has never been accepted. Even WHO says it might be useful. It may be useful. Now coming back to the action, like whatever you have, the anti-inflammatory indicators. You have suggestions, but you are talking as an evidence.
Yes.
And then the mechanism of action, like we are comparing it with this thing undenatured collagen type 2. We have been using it for years together. And I started as an arthroplastic surgeon, basically an arthroplastic surgeon.
Yeah.
But then I actually started off taking more interest last more than a decade now in joint preservations.
Yeah.
So I use them, nutraceuticals and collagen. We have extensive experience with. So if you go into collagen, undenatured type 2 collagen, it is a definite scientific mechanism on the inflammatory cascade, which also basically there, if we talk osteoarthritis only, there's been a paradigm shift for more than 2 decades now. It's not that only wear and tear. It's basically a proper inflammatory arthritis.
Absolutely. I agree, Dr. Vasisht. And with this note, I think we all are very well convinced that not only this molecule has offered a terrific pain relief, early pain relief for acute exacerbation patients, but it has also relieved joint rigidity, improved joint flexibility and mobility to the patient. The initial results, the clinical results which Dr. Dilip Shah had, you had, and John Hunter has shown in his talk, as well as it's been recommended in various international journals of repute. So, Dr. John, your passing remark, what international journals this has been published, and do you think joint mobility and pain relief should be the flagship recommendation, flagship indication for use of this?
Yes. I can send you a list of the publications that we have within our dossier. Now, obviously, a dietary supplement is not going to be featured in the Lancet or in Nature or JAMA or etc., but we have been published in a number of well-respected peer-reviewed journals. And we'll provide you with that list, and you can share that with anybody who would like to see it. Along with you.
This is a great idea for Indian orthopedic trauma surgeons, Indian orthopedic joint replacement surgeons. If we can have this data when we will launch, and we have launched this molecule. So thank you very much, Dr. John Hunter, Dr. Dilip Shah, and Dr. Vasisht for being with me. Over to you, Dr. Diwan, and let's wrap it up. Thank you.
Thank you so much, all these dignitaries who have shared their views on this particular molecule. And I'm sure with this, all the information with Mr. John Hunter has shared, we were quite enriched. And with the endorsement of the usage of this molecule by Dr. Dilip Shah and Dr. Vasisht, I'm sure all of our colleagues will start using this molecule and see good results in their patients. With this, I thank you all. Special thanks to Dr. Shushrut Babulkar for his coordination of this whole show. Thank you very much.
Thank you, and take care, all of you.
Thank you.
Thank you, everyone. Thank you.
All blessings. Bye.
Bye, John.
Bye.
Bye, Shushrut. Bye, Vasisht. Bye. Bye.
Thank you, doctors.